This video is an introduction to the control of gene expression, focused on how this can be brought about by affecting transcription, including the lac operon. With the exception of red blood cells, when you look at any cell in a human body, you'll find that within one particular person, all of the cells contain the same DNA. It's not the case that, say, a nerve cell and a heart muscle cell contain different genes. It's just that in these different types of specialised cell, different genes are turned on and different genes are being transcribed at different levels. Some genes are required by all cells, and we call these housekeeping genes. So these could be things like genes that are necessary to make enzymes for respiration. And then there are other genes which are more specialised, and they're perhaps tissue specific, or they're only required at a particular moment in time. And so they need to be turned on and turned off either during the development of the fetus or possibly at different points throughout the organism's life. It's also possible, as well as turning a gene on or off completely, to slightly upregulate or downregulate the amount of expression in response to various different stimuli in the environment. Gene regulation can take place at various different times throughout the process of protein synthesis, and depending on the particular textbook or revision guide you're using, this may be broken down into 4 different types of regulation or possibly just 3. Neither one of these is favoured in the specification, but you do need to be able to describe what would happen in each one of these stages. So the first point at which we can control gene expression is in transcription, so controlling how much mRNA is made. And this could be as black and white as turning a gene permanently on or off. So for instance, if a gene is never going to need to be expressed because of the tissue it's in, or if you look at the female human body, in any part of the body, one of the X chromosomes will be permanently turned off because otherwise we would have twice as much of those proteins as we need. But it can also be more finely tuned than this. So we could also be talking about up or down regulating the transcription of a particular gene in response to the stimuli in the environment around us. And that often happens by affecting a promoter region. So a promoter region is a small section of DNA, usually just upstream of the gene that it's affecting. And by interacting with this, various proteins or transcription factors can control how frequently that gene is being transcribed. Then we have post-transcriptional control. So this is where something is being done to the mRNA after it's transcribed in order to control how much it's then going to go on and be made into protein. Now, some textbooks and revision guides include translational control as an alternative place where genes may be regulated, and this could involve anything else which is going to stop or start translation or otherwise make it happen more or less frequently. And then finally, we have post-translational control. So this is where a primary peptide has been sequenced and then something is done to that peptide which is going to affect the function of that protein and how much of a particular protein we get. Before we look at the regulation of individual genes by transcription factors, we can look at how transcription can be regulated across the entire cell in one go. For instance, during cell division, transcription stops. And the way that this change is brought about is by changing the form in which the DNA is stored. So you're already quite used to the idea that even though we regularly see chromosomes drawn as being shaped like an X, the vast majority of the time they don't exist in that form. Instead, they're in a form called chromatin. So here we have some histone proteins, and each one of these blobs represents 8 proteins stuck together. The DNA is wrapped around those histone proteins, and where we have those 8 proteins with the DNA wrapped around it, we call that a nucleosome. And you'll sometimes hear these being referred to as being like beads on a string. Now, when the DNA is very, very tightly wound like this is what we call heterochromatin, and this is the form in which the DNA exists during cell division. Because it's so tightly wound and because the histones are so close together, it's not possible for RNA polymerase and also for other transcription factors to interact with that DNA, and that means that transcription will not happen. In contrast to this, during interphase and the rest of the cell cycle, the DNA is in a form called euchromatin, and even though it's still interacting with those histone proteins, it's less tightly wound and there's more spaces between them, and therefore the RNA polymerase can get in there and transcription can happen. Now that makes it sound like we only have 2 options, heterochromatin or euchromatin, and therefore transcription will be switched off or it will be switched on, but actually it's a little bit more complicated than this. Epigenetics is the study of the markers that can be applied to DNA in order to increase or decrease the amount of transcription. So if we look at one of these nucleosomes, as you can see, we have 8 different histone proteins, or rather 4 different pairs. And the DNA is going to be wound around those. Now, part of the reason that happens is because the histones have a positive charge and the DNA has a negative charge, so they're quite attracted to one another. However, we can add a group called an acetyl group, and if you're doing chemistry, you'll recognise it as looking a little bit like this. And the thing with an acetyl group is that it reduces the positive charge of the histones, and therefore it reduces the attraction of the histones to the DNA. And this causes the DNA to be coiled less tightly, which allows RNA polymerase to interact with it more easily, and therefore increases the level of transcription. In contrast to this, another thing that we can add to a histone group is a methyl group. And again, if you're doing chemistry, you'll be very familiar with what that looks like. And what a methyl group does is it increases how hydrophobic they are. And as a result of that, the DNA coils more tightly, and therefore it's even harder for RNA polymerase to interact with it, and transcription will decrease even more. Pause the video and make sure that you can write down the key similarities and differences between heterochromatin and euchromatin, and also the other ways in which the histones can be modified in order to change the level of transcription. So both heterochromatin and euchromatin are made from DNA wrapped around histone proteins, but the difference is in how tightly that DNA has been wrapped. So in heterochromatin, the DNA is really tightly wound, and this means that RNA polymerase can't access it and therefore it's not transcribed, whereas in euchromatin it's loosely wound and therefore transcription is increased. We can modify the histones in 2 ways, either by adding acetyl groups, which will increase transcription, or by adding methyl groups, which will decrease transcription. It's obviously vital, particularly for multicellular organisms, that in addition to being able to control transcription for an entire cell, it's also possible for the organism to upregulate or downregulate the transcription of one particular gene at a time in response to stimuli in the environment or the particular functions of that tissue. So for this we use transcription factors. These are proteins which bind to DNA, and in doing so they either increase or decrease the transcription of a gene that is downstream from there. There are 2 key types. Activators are going to increase the transcription and repressors are going to decrease the transcription. In eukaryotes, these transcription factors will bind to a specific piece of DNA, and if it's an activator transcription factor, then we call that piece of DNA a promoter, and if it's a repressor, then we call it an inhibitor. And usually it's geographically very close to the gene that it's going to control. In prokaryotes, we have a similar system, but using something called an operon, which we'll look at in a second. It's also possible that these transcription factors can be made more or less effective based on their own shape being changed, and this can be affected by binding to lots of other things, including sugars and hormones and all sorts of things. An operon is a region of DNA consisting of several genes and also the regulatory regions that control their transcription. And what's special about them is that they are several different structural genes, so for instance they might code for enzymes, and they are always going to be expressed together, so they can all be under the action of a single promoter. For a really long time we thought that these only existed in prokaryotes, or at least in genes that have come from prokaryotes, so for instance the genes that are found within mitochondria or within chloroplasts. But actually in more recent years we have realised that in rare examples they do exist in eukaryotes too. The reason for doing this is simply that it's more efficient. If you have a series of enzymes which are all needed to metabolize a particular food source, and the only time that you're going to need one of those enzymes, you're also going to need the rest of them, then it just makes more sense to have one promoter to interact with so that you can turn all of those genes on or off at the same time. You need to know the specific details of how the lac operon, which is found in various bacteria including E. coli, works. The lac operon is used by bacteria to turn on a series of 3 genes, which are used to digest lactose when glucose is unavailable. The operon consists of the genes that code for 3 enzymes: beta-galactosidase, lactose permease, and transacetylase, and these are coded for by the LAC-Z, LAC-Y, and LAC-A genes. These are all transcribed in one go to make one really long piece of mRNA, and then split up to make the 3 enzymes afterwards. The first thing that you need to know is that upstream of the genes for the 3 enzymes and also their promoter region, there is a 4th gene, a regulatory gene called LAC-I. And what LAC-I does is it makes a repressor protein. So this protein bonds to a site called the operator, and it kind of overlaps with the promoter. And as long as the repressor protein is there, RNA polymerase is unable to come and bind, and therefore transcription is unable to start. Now, in the presence of lactose, this changes. Lactose interacts with the repressor protein, and when it does, the repressor protein changes shape. And because it's changed shape, it can't bond to the DNA in the same way anymore. This then means that the promoter is available and RNA polymerase is able to come and bind. So therefore the 3 genes can be expressed because they can be transcribed. So when the LAC operon is functional, we do have transcription of the genes for those 3 enzymes, but it's still at quite a low level. In order to maximise the expression of these, we also need a further chemical called Cyclic AMP, and you may have met this earlier in the A-level when you found that it can be made from ATP. Now the CAMP forms a complex with another protein which is called CRP, and these together bind to the DNA. When they do this, this increases the expression of lots of genes, including those involved in the LAC operon. Time for another progress check. Pause the video and without using your notes, write down the answers to these questions. A transcription factor is a protein which controls the rate of transcription, either upregulating it or downregulating it when it binds to a piece of DNA. The 2 types of transcription factor are activators, which increase transcription, and repressors, which decrease it. In eukaryotes, transcription factors bind to promoter or inhibitor regions, and these are usually found directly upstream of the gene whose expression they control. And an operon is a series of genes which are expressed together because they're all under the control of a single promoter. Thank you very much for watching, and I hope you found this a useful introduction into the control of gene expression by transcriptional control. In the next video, we'll look at the post-transcriptional, translational and post-translational control. So don't forget to like and subscribe for notifications for when it arrives.